And so what you see here is that the blue area is what we call the cold pool, which is water that's below 1 to 2 degrees temperature. And you can see in 2010, there was a large cold pool in the summer that went all the way down to the Alaska Peninsula. And then you can see in 2017, the cold pool was a little bit less, but in 2018, it was completely gone from those waters. And 2019 was only a little bit of cold water up there in south of St. Lawrence Island. So why does this matter? It matters because cod historically have been, this is distributed around this cold pool. So on this map, you see the blue as the places where Pacific cod are found during the summer survey. So it's the same map, but this time you're looking at fish distribution. And you can see in 2010 that cod were kind of distributed in the eastern Bering Sea around the outside of that cold pool. But in 2018, all of the cod, most of the cod were found in the northern Bering Sea. And that was the first time we really saw that in our summer survey. And that was very surprising to us. So this has spawned some questions. Like one other question is, are these fish there as a brand new population and they're staying there all year? Or do these fish move out of this area for spawning in the winter? So here are our research questions that we came up with when we first saw this change in distribution. What are the movement patterns of adult cod between winter spawning and summer feeding? What is the movement, number one, what is the movement between the northern and southern Bering Sea cod? Number two, what is the movement of cod between the US and Russian border? Number three, do cod stay under the ice in the winter? And number four, can cod spawn in the northern Bering Sea? And number five was, do cod return to the northern Bering Sea in the following summer? So the tool we use to study this are pop-up satellite archival transmitting tags, or PSATs. And here you can see a picture of Charlotte, who is one of our scientists, and she is holding a cod that we have just tagged. And in that white circle, you see the satellite tag. It goes on the back of the fish with a little harness. And the neat thing about these tags is that they pop up on their own. So you don't ever need to recapture the fish to get information from these tags. And once they pop up, they can give us detailed information on the migration of the fish and on fish behavior. So this study kind of became large pretty fast, and we needed a lot of collaborators and funding sources. And here's a slide to acknowledge all the people and institutions that helped us make these studies possible. And I want to point out, we started out with working with the Norton Sound Economic Development Corporation and with the Submarine Fishermen. That was our very first beginning of this. So here's some of the work that we've been doing up to date. You can see we started in 2019 in the northern Bering Sea in the summer. And since then, we've released tags until 2021 and 2022 in the northern Bering Sea and eastern Bering Sea. And then we also went in the Gulf of Alaska since 2021 and released tags there in the winter. And in the very beginning, we also released tags in the Aleutian Islands in 2019. So this is kind of all the data points where we release tags up to date. What we want to focus on for this talk is the beginning right now is the northern Bering Sea tag releases from 2019 through 2022. With pink and purple marks on the map. Okay, so how did we do this? The satellite tag we're using is by a company called Wildlife Computers. The tag is called a mini-pat tag. It can measure depth, temperature, light, and acceleration, and it's basically a mini computer inside of this tag that you see here on the screen. And it will continuously record those through the entire time the fish is in the water. And then it gets programmed to pop up at any time you give it, about up to a year. And once it pops up, it will give you the GPS location of the pop-up, but it also will give us all the data that were collected with, which help us to estimate the travel paths. And that's the real beauty of these tags.
Is that done through?
And
data?
do you, oh, sorry, go ahead.
We've got
I'm
two
gonna be
questions. We've got two questions. So Adam Bachman here in the audience, you went first and then Orland.
So these fish are moving around and somehow you can tell how long they stayed in Norton Sound or St. Lawrence
Well, we're
Island.
gonna get to that. What
Okay.
all the tags do is record temperature, depth, light, and acceleration. Okay, and when the tag finally pops up, that's when you get the data back. It cannot transmit while the fish is underwater. Once you get those data back, we will feed them into a model, and Julie will talk about that. And that model will help us how to estimate it. So it's kind of like being a detective with circumstantial evidence, trying to come up with the real migration path. Because they're not like a bird where they continuously can beam to a satellite, they're underwater. So all you can do is collect data and then get them beamed up once the tag pops up.
And then, is that answer? And then Orland, you have a question?
Yeah, when the devices do come off, do you guys collect them?
If we can, we do. A lot of times they just get lost. But if we
Okay.
have some come up with the fish, with fishing boats, they get captured. Sometimes they go to shore and we can find them.
Oh,
If we find the tag, we get much better data back because we can basically download it like from a computer. When they beam up their data, a lot of times we only get part of the data because the satellites don't work so well all the time.
Is
okay.
that answer? Okay,
Yeah.
and then you got, okay, and then Katil, you had a question?
Yeah, so how do you define the acceleration and why is that important to record the acceleration?
We have acceleration data. You only get those if the tag is out for less than 90 days, right? And they're used to look at behavior. So you can look at how fast was the fish swimming, how much did it go up and down, all these kind of things, and they give you a clue on how they forage, basically, and how they move, how they swim through the water. So that's a whole nother part of the study I'm not even going to talk about today because it's just too much. But we can answer questions about that separately.
And then there was a good comment to me that it is muffled, this new system that was supposed to be so great. It is a bit muffled. So maybe if we have questions in the audience, you know, those dangling things are the microphones. So maybe we can speak loudly so that everyone can hear us well online as well. Frustrating. New system, everyone. So we're learning to live with it. Thank you.
Okay, so these are just two pictures of how we capture the fish. And that during our summer survey, we released some tags. We caught them with hook and line. And then we went to the village of Savunga and we caught the tags from their local land skiffs with hand lines. And here are just some pictures from our trip to Savunga, so you can see how we did this.
What year was this again?
2019.
2019. Thank you.
So the vessel was fishing for halibut, and while they're bringing up their line, a lot of cod are on the line with the halibut. And we used the cod, we tagged them, as you can see right there on the skiff. We measured them first. We had a little tank set up where they could like put water on them continuously, and then we insert the tag as in the lower picture. And then we release them back into the water with a descender, so the fish go right down to the bottom. Because a lot of times cod have an inflated belly from their swim bladder that puts them right back down to the bottom, and it really helps them to adjust to being back in the water. So when we were in Savunga, we released 8 satellite tag cod and 75 cod with conventional tags, which are little plastic we call them spaghetti tags with a tag number on it. Okay, so here are the pop-up locations. From the year 2019, we released 38 tags. We heard back from 33 of them, which is pretty amazing. And 6 of those were caught in the fishery. So those, we actually got the tags back. The fish were brought up by fishing boats. We had not a single fish die of tag mortality, which was pretty amazing. And we could do this geolocation, which is basically establish the movement path with our model for 31 of those fish. And now I'm going to give the stage to Julie, who is the master modeler for this.
Okay, thank you very much, Suzanne, and thanks, everybody. As Suzanne mentioned earlier, it's like detective work. So we know where the fish was released, and we know where it pops up, and then we're using the data from the tags as our clues to figure out where it went. And what we're going to be doing is reconstructing the path, but what it looks like is a location each day with our estimate of error. Because we don't know exactly where it is along that way. So that's what we're going to be doing with the satellite tag data. And the major clues that we're using come from the depth and the light sensors on the tag. So on the top here, this is an example of just 5 days or so of depth readings. And what you would do is you would say, oh, okay, so this the maximum daily depth, you assume it's on or close to the seafloor. And then you can match that depth, so say 80 meters, to your bathymetry map and find all those grid cells that are close to 80 meters. So that's kind of how you link the depth data from the tag to the maps in the study area. The second thing that we use are the light sensors of the tag. They measure the increase in light intensity during the day and drop off at night. So you can then use that to get the time of local noon, which is longitude, and the time of sunrise and sunset, which is latitude. Longitude is a lot easier and to estimate with this, so we use that mostly, but sometimes we use latitude as well. So those are our major clues from the tags. Then we put it into a fancy Statespace model. Actually, it's not that fancy, it's pretty simple. It was developed by Martin Peterson for Atlantic cod, and then I just adapted it for Pacific cod, and that's a paper that's just recently come out in biotelemetry. And I'm not going to get into the details of this, but you can please feel free to ask me later. But basically what you're doing is you're dividing the study area up into grid cells. And then ultimately what it tells you is the probability that the fish occupied each grid cell on each day that there was data. And this is an example of what it looks like. So this is basically, it's a probability surface for one fish on one day. And so the highest probability is shown here in this lighter color, but it could have been anywhere within this grid surface area. So you've got that probability surface, and you can do things like construct polygons around different levels of that probability. So you can have a polygon that holds the highest 99% or the highest 50%. And then you can also find that point that's the most probable on each day. So those are just some ways that we can describe and talk about the pathways that we are reconstructing here. Then you can take those probability surfaces and combine them. Like for one tag, you can look at different periods of time, like say, what's it doing over a month or during a certain period of time. And then you can also take probability surfaces for multiple animals and get an idea of the location probability for all of your tagged animals combined. And so you can look at them monthly, during the spawning period, etc., or during summer foraging. So this is an example of what a reconstructed pathway looks like. So this fish was tagged in the Northern Bering Sea in September 2019. And what you've got is its daily locations color-coded by month. And you've got your 50% is the darker gray and your 99% the lighter gray. So this is the error in our estimate of its location. But this fish, the pop-up was in the Northern Bering Sea to June the following year. So if you didn't have the satellite tag data, you would think, maybe it didn't move very far, only 250 kilometers. But with that reconstructed pathway, you can see that it actually did move quite a distance down to the Pribilof Canyon area during the peak spawning time in March. And then it moved back to the Northern Bering Sea following that. So it actually moved something like more like 1,500 kilometers round trip. Then I'm going to show you how they all move together. So these are composite probability slides, and the number changes for each month And so keep track of that as I kind of cruise through what all of our tagged animals are doing for each month. And the highest probability is again, the lighter color, the yellow here. So.
So if I can just interrupt, so the yellow in your model of where the fish went, the yellow is really where you think it went, and then.
So yeah, so most of the probability is in this area where it's lighter colors. So this most of the tagged fish are probably in this area,
Yeah,
but they could be anywhere within this extent.
okay,
And that's from 24 animals.
okay.
So there's animals over here, a lot more animals over here. Does that explain it?
Yep, that explains. So yellow is more likely where they are, and
Yeah,
blue or purple or whatever that color is,
still
the darker the
probable,
color, it could be in there too.
could be in there as well.
Thank you.
So that's August of 2019, and here's September. And October, so they're basically spreading out within the tagging area, within the northern Bering Sea. And just heading out just slightly in November. And then, but some are heading south already, and then in December, here's the sea ice coming in with this hatched line right here. So starting in December, the fish all start to move to the south and the west ahead of the oncoming sea ice. So here's January and February and March. And so by this time they're all in the shelf area. Then now we're going to look at their distribution during the peak spawning time. So this is mid-February to the end of March. And they're all along the shelf. And I put in the actual pathways that got them there. And these are, remember, these were where they were tagged in the northern Bering Sea. They moved to the canyons and slopes. They moved 400 to 1,000 kilometers to reach these areas. And most of the composite probability was in the eastern Bering Sea during this time. There was some probability in Russia and some probability in the Gulf of Alaska, but almost none in the northern Bering Sea itself where they were tagged. And if you line this up with what we know about spawning locations from actual capture of fish in spawning condition, this is work by Sandy Neidiger, you can see that our distribution of the tagged animals during this time actually matches up pretty well with this. Then another way of looking at this location probability is by month. So here we have month on the x-axis and the location probability for the entire study area on the y, and by region how it changes. So you can see that it starts off with most of the probability in the northern Bering Sea. It declines starting in November as the probability in the eastern Bering Sea rises. Note that there's some probability in Russia year-round, and there's some in March in the Gulf of Alaska during the spawning period. And then our sample size really drops off in the summer months. But there is probability of return back to the northern Bering Sea. We can use the distance that they move from their release location in a model to estimate the timing. So when did they start their migration? So you're looking for the inflection point of this curve, basically in this model. And what we found is that the start of the migration was middle of November plus or minus 10 days. So you might think, what does that have to do with temperature in the study area? Well, this is temperature data from 2 stationary data loggers that we put. This black one is in Sabunga, and the lighter gray is Western St. Lawrence. And so we're measuring temperature over the whole time period here. This blue line is our estimated migration onset date. And just note that that occurs in conjunction with the steep drop in temperatures in the study area. And then you can see when the tag gets covered with sea ice, everything's minus 1.8 until the beginning of May. The tags also contain, in addition to the geolocation, they have depth and temperature records that you can use to learn about what the fish experienced during that time. And so this is the maximum daily depth of all tag fish. So the actual individual observations are in gray here, and the mean for all fish is in the red. And so what you can see is that during the foraging, so here's our migration start date in November. What you can see is during the foraging period of time, they're mostly in the shallow waters, less than 50 meters of the northern Bering Sea. And then during the migrating, they start the depth gets deeper. And then during spawning, it's between 200 meters and around 100 meters. And then it gets shallow again as the fish, of course, our sample size drops off in the in the spring, but those fish did get shallower. Then, excuse me,
There's water underneath right here.
Oh, there's-
There's a black cup of
excuse
water.
me. Okay, so then temperature. This is the maximum daily temperature. Again, these are from individual fish, and then the minimum is in blue. And so that foraging period, you can see that the fish experience temperatures all the way from more than 10 degrees and less than 0. So it was a big range of temperatures that the fish experienced during that foraging time in the northern Bering Sea. Then the temperatures start to get colder as winter's coming on and they're migrating. But then during spawning, the temperatures increase again as they go to places that are warmer to spawn. And this is likely related to, the optimal temperature during spawning is going to be 3 degrees to 6 degrees for the survival of the larvae they're finding. So it might have something to do with that. And then it gets cold after spawning as they begin their return migrations. And then warm again as you get to the following summer. Okay, so that was 2019. In 2021, we released 18 fish, and this was only from the survey vessels this year. And their release locations are these white triangles. And 17 out of those reported, we had mortality this time. We had 4 animals that didn't survive the tagging process. But of the fish that did, the tags that did pop up, we had 8 of them that happened during the summer months. So we got information about their activity and locations during the summer months. And then 3 of them were in the migration period here in December. And then we had 1 observation from March during spawning. And this is just arrows that kind of link where everybody moved to. So it's pretty similar to 2019 during the summer. Didn't move very far, always mostly within the northern Bering Sea and some up into Russia. But what I want to draw your attention to are these 2 fish. This is Popup's 365 days after the fish was tagged. So again, if you didn't have the satellite tag, you might think, they didn't move that far. But when we can reconstruct the movement of these fish, you can see that, okay, this fish started, it was tagged right here. According to the model, it went up north and then moved back down through December, October, and then spent the prime spawning just north of the peninsula, and then made its way back up into the northern Bering Sea, where it popped off at the end of July. Then the other one, so this one, the release location was almost exactly near the Popup location. But this one, again, moved south during the winter to almost the same location as the other one and moved back to where the tag popped up. So again, quite a long distance movement, which you would not have known except for these satellite tags. And this is putting those 2 trajectories together with the pop-up locations. And you can see that these green of the reconstructed pathways and the pop-up locations matches very nicely. And all along, they're very, I did the processing for these at different times, but they're remarkably similar to me. So some pretty cool stuff going on with annual movement patterns for these. And in 2022, we had fewer tags out. We had 6. 4 of those 6 reported, and of those that did, 1 was in November. They moved very little, but the other 3 went all the way down to the northern part of the peninsula during spawning. So again, big movements out of the northern Bering Sea. I haven't done the reconstruction of these pathways yet, but when I do, we'll get more information on migration timing, etc. So overall, for all those 3 years of tagging, we have no evidence of any cod, any live cod overwintering in the northern Bering Sea. We would know that from the location and from the temperature records. And we noticed that migration onset begins with cooling temperatures in the study area. The tag fish, when they moved out ahead of the sea ice. And most of them stayed ahead of that, the extent of the sea ice. We found substantial seasonal connectivity with the eastern Bering Sea, where they moved to traditional spawning areas. And we found some seasonal connectivity with the Gulf of Alaska and some connectivity with Russia year-round. And also some evidence of summer cyclodility, so fish returning to the northern Bering Sea after they've left to go spawn elsewhere in the winter. And now I'm going to turn it back over to Susanne. So that was talking from releases in the summer. Now Suzanne's going to be talking about releases in the winter.
So is there any questions so far? Yes.
Do the cod spawn in the water column, or do they spawn hard on the bottom? Is there a reason they go to those deeper depths? Is it structure?
I think, yeah, I think there's a lot to where they spawn. They're very particular. It's kind of our study in the winter where we went with a local fisherman. He knows all their spawning spots. They all like to spawn on the edge somewhere. And they like to spawn deeper than they are during the summer, between 100 and 200 meters, seems like to me. The Bering Sea cod spawn off the shelf edge. And there is warmer water deeper. That, you know, the deeper you go, you're down to your 4-degree, which is the most dense water. So we found a scientist in Oregon, Ben Laurel, has done studies on larval survival, and they found that cod survive the best between 4 and 6 degrees. So I think they find that depth. They do spawn close to the bottom, maybe in the water column. We haven't really seen them spawn. And their eggs they fall to the bottom, but they don't stick to the bottom. And then it's kind of complicated as they develop, they change their density, and then I don't know all the details, but they have found that the larvae are then in different parts of the water column. So it's kind of important where they are. And we don't understand it all. I don't understand it all. Yes.
So I have a question. This is Kay. Did you get your answer? Okay. So my ignorance, I'm not a expert in the Pacific cod, but obviously you guys knew a lot about the southern Bering Sea Pacific cod. That's living down and getting caught by the commercial fishermen in the big fishery. So it sounded like you guys were kind of, wow, those big long travels. Is the northern, what was most surprising about that to you? That these are fish that were from the southern Bering Sea and kind of remain loyal to their spawning grounds, or what was the big take home to that? Or are we seeing southern Bering Sea fish? Were these northern Bering Sea fish that were here to begin with, or were they southern Bering Sea fish acting differently? Was that?
Yeah, but there's a whole nother part that I'm not even going to talk about today, which is genetics.
Okay.
We have done genetics, and the genetics are showing interesting patterns. And there seems to be, just to say in a nutshell, there's like a Russian, what we call Russian northern Bering Sea stock.
Okay.
That spawns in the northern part of the eastern Bering Sea, just south of that border. Let me go back to a map. You see this border here, they're spawning in this corner here. And that stock seems to be different and that stock moves, of course, in between those two. That seems to be different from stocks that spawn in this area here. And they're even different from stocks in the Gulf of Alaska and here.
So that area you were circling west of St. Matthew, you would call that Eastern Bering Sea?
Technically, as a management area, it's the southeastern Bering Sea. But this is where the management area draws the boundary.
right here,
Okay,
okay? And then, of course, Russia is right over here.
yeah,
So the
Anna, dear,
genetics are complicated. And I don't want to get into it
Okay.
all, but we are working on it and there is patterns.
yeah, it's just you have
They
a
are
whole
different.
different water body over that far, west. West of
Yes.
St. Matthew. That's why I would think it would be a different name, but
So
thank you.
they spawn in the they like the canyons.
Yeah.
Yeah.
All right, thank you very much.
So what, to me, one of the most interesting things is these fish that we tagged in the Gulf. Moving all the way up. And that's kind of what I'm gonna talk about here next is we went in the Gulf of Alaska. There was a big population crash during the really warm years, during the blob years. And people wanted to know, did fish die or did some of the fish move into the Bering Sea? And what is the connectivity between, especially the Western Gulf cod and the Bering Sea? So we went, worked together with the Aleutianese Borough. We chartered this vessel, fishing vessel Decision, and we captured the fish with pots, brought them to the surface and released them. And this is what we saw. So the first year, this is now the fish in the summer. We released the fish in the yellow dots in the winter. And now this is the probability of where these fish are in the summer. Only 34% of fish stayed in the Gulf of Alaska in 2021. And the rest moved north, and we had one that moved all the way through the Bering Straits, all the way up here. That kind of blew our mind a little bit. And so we went, the next year in 2022, we tagged fish again in the Western Gulf. So what you see here are the travel paths from 2021. And then here in 2022, only 5 out of 25 fish moved into the outside of the Gulf of Alaska, and the rest stayed in the Gulf. So a much smaller percentage of the fish moved, which is the red dots, in 2022. So what's going on? We wanted to know is what was different between those 2 years. And so what you can see here is the Pacific cod distribution in the summer survey from 2019 through 2022. In 2019 is that warm year that we saw this big movement into the northern Bering Sea, which is that pink circle. 2021, it was still warm, but not quite as warm, and the fish stayed a little bit further south, like south of St. Lawrence, St. Matt's area. And then in 2022 in the summer, it was much colder. And we had suddenly, again, a larger distribution of cod in that corner here by the north of the peninsula, and the fish in general just stayed further south. So the fish were in different places in 2022, and we saw that in the tagged fish from the Gulf because it was colder, the water was colder.
The one.
Yeah.
The one fish that made it up to Point Hope, that 1%er, did it pop up there? Or?
Yep.
Yeah, we know it was there.
Yeah,
It was very surprising. So we know, yes.
but it's
What's the?
always been in my mind, do the fish that go past the strait? Have we ever seen any movement back of?
Yeah. You don't know.
Well, so it popped up and then we don't have any way of knowing. So it's just one fish.
Cause you're implying that that fish has to swim against the river, which is the northbound water going through the
So
strait.
it might have been.
I mean, those are, that water past Diomede is going sometimes 5 knots. So if those, it's a good question on whether or not.
Are they lost to the system? Is that kind of what you're saying? Once they're
Yeah,
north?
and I- Russians over there pollock fishing or were for a couple of years. I don't know what the status is now
They're currently looking at, bring that up later.
on the other Bering Strait. So thank you.
Yeah, so for summary for the Gulf releases is that we did see connectivity with the Bering Sea all the way to the Chukchi Sea. And that we saw that the extent of the migration and the proportion of fish that migrate might vary with different years and different temperatures. So here,
kind of wrapping it all up, we're coming back to our research question. The first question was, what is the movement between northern Bering Sea and other management areas? And we've seen this now, that there is quite a big connectivity between the eastern Bering Sea and northern Bering Sea, and between the Gulf of Alaska and the Bering Sea, and all the way between the Gulf of Alaska to Russia and the Arctic. What we did not see was any kind of movement between the Aleutian Islands and the Bering Sea and the Gulf. So the Aleutians seem to be kind of their own system that don't really much interact with the Gulf and the Bering Sea.
Are there cod in the Aleutians?
Yep, there's quite a bit of cod, and we just don't see a big exchange. Like the cod we tagged there stayed within the Aleutians.
Did you see any genetic differences?
Yeah, and they're genetically different. I'm sure there's some mixing, you know, around Unimak area and stuff, but in general, they have a different genetic signature, and there just doesn't seem a huge exchange like we've seen with the other areas. The second question was, do cod stay under the ice in the winter in the northern Bering Sea? And we did not see any live fish under the sea ice. So we think it's unlikely that they can stay there and spawn. But, you know, we can't exclude it all the way. Can cod spawn successfully in the northern Bering Sea? We have not seen evidence of that. To date, all tag cod moved out of the northern Bering Sea during the spawning season, but I want to say it's probably related to the area being covered with ice. I do not know what would happen if there is no ice, and because they do spawn on the ice edge. So, I guess we can just see what happens in the future with that. Okay.
Vera Metcalf has a question.
Yeah.
Vera, we see you. Go ahead.
Hi, I'm Vera Metcalf. I'm born and raised in Savunga. I'm glad that you will be out there to provide the reports. I was wondering, you know, I really support indigenous knowledge. So I was wondering, do cod winter in, or you know, have wintering thing? I was wondering if you could ask that question when you're out there in Savunga or Gamble about that question, because you know, maybe indigenous knowledge can provide a different perspective.
Yeah, I'm very glad you mentioned this. I think when we were there last, we talked about you know, the hunt in the winter and access, and also winter ice fishing. And I was very interested to see if anyone captured any cod or had seen anything like that. And I just don't think we ever got into it enough to learn from a lot of people. I just talked to the people that I interacted with at the plant. That's a great point. Yeah.
You might want to mention the public meeting on the 24th.
Yeah, so we have a meeting on the 24th in Savunga, where we're gonna give kind of a version of this talk, and we can hopefully talk about this kind of stuff then. That would be great.
Wonderful, thank you.
And to follow up on Vera's good point, Vera, thank you. Is there a component of this study that does not just, you know, ask during a meeting, but actually record or have a contribution by people, because you're with honestly, you're with some of the most proficient and knowledgeable people of the ecosystem when you're working with St. Lawrence Island, residents of St. Lawrence Island. So there's tremendous, when I've traveled out there on the topic of Pacific cod, there's tremendous history, stories from the 40s of cod, and especially towards the eastern side. So I think there is a lot of information, and I don't know how your study is designed, but I would encourage as you go forward to maybe put a whole some time and effort into making sure you're talking to the historians and of some of the resources out there, because there is tremendous amount of knowledge, I'm sure, about cod. And I think you would learn a lot, or have new questions maybe even to think about, both timescales and movements. I know that just putting this poster out on Facebook for this talk, there was people wrote in with their own stories of catching cod in the spring on the edge of the ice. So the cod were there in the spring, and that people near Diomede would catch boatloads of cod in the spring, you know, fill up a boat. So, you know, what is that? When was that? And what years? Those kind of things might really
It's a very good
give
point.
you some glimpses. If you have the privilege to work with the peoples of the island, I recommend somehow incorporating
Yeah.
that.
Yeah, we
Well, Orland, go ahead.
we go around maybe March, February, March, around there, April, springtime. We go to the other side of the mountain here, and we go cod fishing, and we do catch them, the tom
Oh, I would
cod.
love to hear about that, yes, and to know where that is. Cause that's their spawning time.
Oh, okay. Spawning
Yeah,
time.
that would be very important to know, because we don't know anything about winter distribution. We don't have anybody looking for cod in the winter in your area.
Oh,
But
maybe
it's
I could record next time. I maybe go
wonderful.
ice
Yeah.
fishing. Nice.
Thank you, Orland.
Yep.
I saw the first, call it first contact, was like late 1800s, a photo of 3 fishermen in Gamble they had sticks with cod that were like the lengths of their body. It was like, yeah, cod's been here for a long time.
Mhm,
Yeah.
mhm.
It just would, I biologically, there has to be a zone in which cod can feed and digest. And that kind of helps them in their range. And do we know what, looking at your foraging, it was like, it could be this cold or it could be this warm. Is that data that we know already, or?
We know that cod can swim through really cold water. We also know that they don't, like when we look at them in the summer, they tend to not be in the cold pool. They tend to be in warmer water, but it's not a physiological barrier. They can be in there, they just choose not to be in there. And it most likely has to do with food, I would think, because where cod are in the summer is where they're eating. We also have speculated that there could be a northern stock of cod that is more resistant to cold water, that developed with, you know, abilities to resist cold water, and that the more southern cod might not have that. And there's actually a gene in there that makes their blood have that antifreeze for other fish. And so we're kind of looking at that to see if that's maybe something that the northern cod have, but a lot of that we don't know. I think people just have not really focused on cod in the northern Bering Sea. You know, to know that. So,
When you have been tagging cod, have you been targeting certain size groups, like the bigger, more mature fish, or I would assume that you have a little bit different distribution of the young fish compared to the mature fish. That's the case with Atlantic cod, and I could be the similar with Pacific cod too. Like
yeah, for sure.
Atlantic cod, the younger cod will be more on the eastern side of the Bering Sea, and more mature would be more towards the western side.
Yes, so that's exactly what we find in our summer surveys. The smaller fish are more on the shelf, and the larger fish are on the edge of the shelf, where most of the fishery happens in the summer. There's definitely a pattern of size distribution in the summer that we see from our survey. Yeah.
I'm done. Thanks. And so this is Gay, and I'm just thinking you were tagging the fish on the northern side, central northern side of St. Lawrence Island. And it looked like from your map, they were kind of going down both sides, maybe.
Mhm.
I was kind of interested, your yellow in Julie's presentation, that yellow stayed pretty far north for a very long time, actually. I mean, you were showing some fish drop down,
Yeah.
but some fish didn't
Some
go.
fish didn't.
Have you thought about maybe next steps, once you've done the cod or Savunga, cod off Savunga, maybe enhance your study and maybe go west? Because you'd probably be getting into that Russian group, and it looked like you had some fish that weren't afraid to stay into November. And when you were saying Russian, but not northern Bering Sea, if you look at the latitude of that, that is the northern Bering Sea, where that, you had a slide back for the winter time, and I was like, isn't that the northern Bering Sea? You know,
Locked up.
oh no,
It's not locked now.
sorry to
So,
make everybody go back.
are you talking about this area here, right here?
Yeah, I think if you go back to where you had the purples and the yellows and all of Julie's,
Yeah, that's the way to do it.
yeah, otherwise we'll kind of have someone goes into a seizure.
Like
Right
here,
in there, if you go to like one of the later fall,
November?
yeah, look at November and then go to December, see, they're all over there between
Right
Chukotka and right on the border, look, you can see the line
Mhm.
right through it, up there west of Gamble. And then go, that's November, and then
Mhm.
go to your December, it struck me. Because that is, to me, that I'm looking at that and I'm thinking, well, yellow is the truest movement. That is northern Bering Sea, and that is December, isn't it? Look at that bright yellow right there. And I
Yes,
get it, there are other fish that are peeling off going
that's right.
east.
So, yeah, you can see like the individual blobs are like individual fish.
Right, so that bright yellow really caught my eye, because I get it, your eyes going down to the eastern Bering Sea, but my eye was stuck up here.
Yeah,
That's northern Bering Sea for sure, if that bright yellow, look at where Gamble, the west side of St. Lawrence Island is Gulf of Onadir. Ooh, and those were fish that were taken from mid-center. So what would it look like if you had fish captured in that bright yellow spot? You know, it made me wonder if that wouldn't push you into, you know, Navarin, Cape Navarin, southern edge of the Gulf of Onadir, or into Onadir. Anyway, thank you for going back to that, because I was like, I think that is northern Bering Sea in December.
oh
But that
yeah,
would be
in
a-
December, but then by March, it's all out of the northern Bering Sea.
Yep.
See, what happens is the ice happened, and then they all kind of stayed ahead of
But
the
see how
ice.
north they are still. They're still
are,
north of
Yes.
Onadir. Those
then-
bright yellows.
Then in February, and here we are, this is February, March.
Still in the
so we
northern
had,
Bering
we
Sea.
had definitely some fish in here, but.
See
yeah.
that northern Bering Sea kind of under the ice, it's
Yes.
bright yellow. So
Yes.
somebody stayed in there, but that's just me. Anyway, that was my
Well, that
notice.
it's kind of hard to see here, but this was early, what is this, early February? There was an opening in the ice, and then late February it closed up. So if
Okay.
you look at all of February, looks like they're under the ice, but they really weren't.
I
They
see.
just stayed ahead of it. They were kind of pushed up by it. But it seemed to me, you're absolutely right. The fish stayed until they really had to leave, because now here's the ice.
And those
so
are
they- Yeah.
these fish
Yeah,
west.
Mhm.
I think that's different water maybe, or.
And so we're seeing like these fish are looking genetically different
Okay, thank you.
than these fish. It's really interesting. It falls out like that, but that's a whole nother study to talk about that, we just didn't have time
And
today.
in the chat, I just wanted to add, and Vera, I just saw yours, so I'll go Rick Toman, then Vera's comments. So Rick Toman says, fall, early winter 2019-2020 was a late ice arrival around St. Lawrence Island. Well, thank you, Rick. And then Vera had written Ukok in Yupik, if I say that correctly, I hope. It's been a long time, it's been around a long time, cod. And Orland agrees with Rick Toman, that was the year it was warmer.
Mhm. It was a really warm year, I
Yeah.
remember that, yeah.
Thank you all for the chat.
Okay, so all I wanted to do is acknowledge people in the last slide and say thank you to everyone. Let's do this. So these are,
Tim Lower has his hand up, go ahead. I say that right, Lower?
Yep, you got it.
Go ahead, Tim.
Yep, you said that right. I'm probably throwing in an entirely different question, but especially with those maps, I was wondering, do Arctic cod get that far south? Is there some overlap between Arctic and Pacific cod with that ice edge movement?
I do not know this, Tim.
Okay,
Are
good
you
enough.
talking about Arctic cod that are related or similar to tom cod? The really small foraging fish from the Chukchi Sea?
Yeah, they're kind of skinnier and mushier, if I recall correctly. But yeah, they come down from the Chukchi.
We get them in Nome, I don't know
Okay.
how far south they go.
To get that information though, I would recommend going to the NOAA Race Division Bottom Trawl Survey website, because they have some great reports, and they are every 20 nautical miles taking a look at the complete Eastern Bering Sea and Northern Bering Sea, and they have those numbers presented for several
Yeah,
years.
If the Arctic cod are getting pushed down there, I would expect it to happen with the ice edge in winter when they're not.
Yeah,
Surveying.
so I don't know if we have winter data from those cod species in the Northern Bering Sea, unfortunately.
Okay,
But
good
I
enough.
say, I remember Ben Laurel and I talking, and he did say that Arctic cod can be under the ice. They have that antifreeze in their system compared to what we think of Pollock and Pacific cod.
Thanks.
Thank you.
All right.
Okay.
Well, thank you, everybody.
All
Thank
right.
you for all the questions. That was great.
And thanks everyone, and this is the time we ask that people throw a little love in the chat box for Julie and Suzanne. Like I say, I know, speaking from experience, it's never easy to be a public speaker. You guys should get right back up there, or I will. It's never easy to be a public speaker and to bring, they're headed out, so you're headed out tomorrow. And what is your plans? How many fish are you gonna try to tag and?
We have 23 satellite tags in my little suitcase.
Okay,
So hopefully we get those all out. And then another interesting thing we're gonna try to do is put out little baited cameras for the baby cod. To see if we can find young of the year cod close to shore. Cause if we do that, that would indicate they're actually spawning
all
in
right.
that area. Cause you don't find those baby cod very far from where they were spawned.
And then how long are you guys planning on being out in the Savunga St. Lawrence Island area?
We will be there for a week.
Awesome. Well, we hope you have much success. Who will you be working with? Is this with the out at Savunga? Is this the NSEDC or the Native Village of Savunga?
We have been working with NSEDC to set this all up, and that's been wonderful, super great support. And we will working with the plant in Savunga and the tribal fishermen to, we chartered 2 vessels, and they were the same fishermen that were out there with us last year.
Okay.
We're very excited to work with them again. It was a great experience. And so, yeah, hopefully it will all work out for us.
And have you been out there before?
No, I have not.
Okay.
It's my first
all right.
trip to the
Julie's
area.
First trip to the Bering Strait region,
Yes.
so.
Yeah.
Well
I'm
done.
excited.
So, thank you all so much. Any other questions? And I'm away from my computer, so the chat, go Adam.
Do we have communications with Atlantic cod fishermen? Have we ever seen any Atlantic cod on this side of the world? And have they seen any Pacific cod on
On
that
their
side?
side of the world.
And we're looking at this other ways to try to figure out whether there's any kind of migration either way.
I haven't heard of any of that.
I'm just
Right.
wondering if that would be a good question to ask.
Well, definitely, there's
the Atlantic cod, we were just talking about this earlier today, Atlantic cod is well studied and they know vastly, they know a lot more about the movements and genetics and population structure and so much more about Atlantic cod than we know about Pacific cod. And finding out those what's might be similar would maybe help us just have standpoints for asking more questions or.
Because
sort
that
of
cod
thing.
that went from the Central Gulf to Point Hope had made it more than halfway to the Atlantic.
No kidding,
They-
yeah.
Yeah, so they move long distances, and I know that Atlantic cod can do that too. They can be big movers and.
So
Yeah.
2 questions. We're going into a warm spell the next 2 years, they tell us. Are you guys gonna be tagging your fish?
We're
in the next
hoping
2
to
years.
do
You're
that,
hoping to
yeah.
do that?
We got a very nice, a big grant from the disaster relief fund for the Gulf of Alaska cod, so we can, and in that grant, we also asked to tag in the Bering Sea.
Okay,
So we have money for at least next year to release more tags, and then hopefully we can continue.
well, buckle in. We're all kind of
Yes.
buckling in, wonder what we're gonna get
Yes.
here.
Yeah.
And then the second question, last question is, will you come back to Strait Science and tell us what you found from your tagging this year? Would be very interested to know after
We'd
this.
love to come back,
All right,
always
Yeah.
love to come, back.
awesome.
yes.
Thank you very much for hosting.
You bet. And so know that for everybody, the next Strait Science is not going to be until September 14th, and we're gonna have Tim Fowle. And it's gonna be, he's talking about the Port of Nome and the consortium of scientists that are coming to do a lot of work looking at the Port of Nome development. So that'll be very interesting to hear about all the research that's coming, specifically to ask questions about the port, and that'll be September 14th. The reason Strait Science is sort of less right now is because of the harmful algal bloom issue that's ongoing in the region. We have no new update at this time because we have no big study projects underway after last year's excitement. However, be safe, Bering Strait. This is the time of year last year that we had some of the highest numbers of the saxitoxins. So if you feel sick eating seafoods, don't stop eating your native foods. But if you start to feel sick or have some strange feelings or symptoms, please go see your healthcare providers immediately. Could be someone didn't wash their hands when making the potato salad. I've had that happen to myself. Or it could be something going on with the foods. It's always good to, if you have something not right, go find out what's going on. So with that, when October rolls around, we have a full lineup. So we're going to be going great guns starting again in October, but the next Strait Science will be September 14th. We hope to see you there, and go ahead, Orlin. Go ahead.
No, I was just saying, sounds good.
Okay. All right, with that, everybody have a good night. We'll see you next time.